artificial intelligence has taken another important step in the field of biology. Research teams, including those from the Arc Institute, recently released Evo2, a large language model trained on 9 trillion genomic sequences. Now let's take a look at what makes this model special. This video will share the Evo2 paper with everyone. The most striking aspect of this model is that it is the first fully open-source large model for biological sequences, including training code, inference code, data, and model parameters, all of which have been made public. The biggest feature of Evo2 is its versatility that it can handle various biological genome sequences from bacteria to humans. Specifically, the model has demonstrated 3 main capabilities. First is the sequence generation capability. Evo2 demonstrates powerful DNA sequence generation capabilities, capable of generating various types of genomic sequences, from simple mitochondrial genomes to complex eukaryotic chromosomes. This generation capability spans across different life forms, including prokaryotic bacterial genomes and eukaryotic yeast chromosomes. Taking the generation of yeast chromosomes as an example, the difficulty and complexity of this task are quite high. The model only needs to be given a 10.5 kb, 10,500 base pairs, sequence as a prompt, and it can generate a continuous sequence as long as 330 kb, 330,000 base pairs. This long sequence generation capability is quite rare in biological sequence models. Even more surprising is that the quality of the generated sequences is also quite good. The sequences generated by the model contain several key features of eukaryotic genomes, including fully functional tRNA genes, important molecules responsible for translating genetic code into proteins, promoter regions capable of regulating gene expression, and protein-coding genes with intron-exon structures. Although the density of these features in the generated sequences is lower than in natural genomes, it is already a very significant advancement. From a functional perspective, the DNA sequences generated by the model also exhibit good characteristics. That it is important to know that one of the most important functions of DNA sequences is to encode proteins. The genetic code in DNA is read by cells and used to produce proteins. Researchers found through computer analysis that if the DNA sequences generated by Evo2 were translated into proteins, the structure and function of these proteins are similar to those found in nature. This discovery is very important because it shows that Evo2 has not only learned what DNA sequences should look like, but also understood the core mechanism of life, how DNA sequences encode functional proteins. Next is the predictive ability of mutation effects. In biology, a mutation in a DNA sequence refers to a change in the basis of the sequence. This change may affect the function of the organism. Evo2 can predict the impact of these mutations. Specifically, Evo2 can predict 3 important types of mutation effects. First, the impact on protein function. When a part of the DNA sequence that encodes a protein changes, it may lead to alterations in the protein structure or function. Second, the impact on RNA function. RNA is the messenger molecule of DNA and plays a crucial role in cellular functions, and mutations in its sequence can also lead to functional changes. Third, the impact on biological fitness, that is, whether these mutations will affect the organism's survival and reproductive capabilities. Traditionally, predicting the pathogenicity of variants usually requires sequence alignment, which involve aligning and comparing similar sequences from multiple species to see if a variation at a certain position is evolutionarily conserved. However, Evo2 is the first language model that can accurately predict pathogenicity without the need for such sequence alignment. What is even more surprising is that Evo2 can not only predict single base changes, called single nucleotide variants, but also predict more complex types of variants, such as DNA fragment insertions, insertion, or deletion. This comprehensive predictive capability makes Evo2 a powerful tool for clinical variant analysis. The third is the ability to design the epigenome. Here, it is necessary to first explain what the epigenome is. That we all know that the genome is composed of DNA sequences, but in cells, DNA is not simply scattered, it is packaged into different states. The regulation of this DNA packaging state constitutes the epigenome. An important feature of the epigenome is chromatin accessibility. Chromatin is a complex of DNA and proteins, which can be in an open or compressed state. When chromatin is in an open state, this DNA is more easily accessible to proteins in the cell, making it easier to be read and expressed. Conversely, when chromatin is in a compressed state, this DNA is less accessible and less likely to be expressed. The research team developed a method based on inference time search, which can design DNA sequences with specific chromatin accessibility patterns that, in simple terms, they can predict and design certain regions to be in an open state by controlling the characteristics of the DNA sequence, while other regions will be in a compressed state. This capability means we can precisely regulate gene expression by designing DNA sequences. To demonstrate the versatility and precision of this design capability, the researchers conducted an interesting experiment. They encoded Morse code messages in the epigenome. The specific approach was to design DNA sequences in such a pattern. Narrow open regions correspond to dots in Morse code. Wide open regions correspond to dashes, and inaccessible regions correspond to spaces. The research team successfully encoded several simple messages, including LO, the first message transmitted on the internet, and the first word in Edmund Spenser's The Fairy Queen. ARC, the name of the institute conducting this research, and Evo2, the name of the model. These diverse messages were successfully encoded into DNA sequences, demonstrating the reliability of this design method. To understand how Evo2 works, researchers used a sparse autoencoder to perform an interpretability analysis on the model. Interestingly, even without any biological annotations, the model learned features related to various biological elements, such as exons, introns, and transcription factor binding sites. This indicates that the model has indeed grasped the biological rules within DNA sequences. Of course, Evo2 also has some limitations. For example, during sequence generation, the feature density is lower than that of natural genomes. Researchers believe this could be addressed by optimizing inference strategies or improving the model. Additionally, although the generated sequences may not have significant sequence similarity to natural sequences, they're still semantically valid and may retain functionality. It is worth mentioning that considering the safety issues of biotechnology, the research team conducted preventive assessments and risk mitigation before the open source release, demonstrating a responsible attitude towards artificial intelligence research at the same time. The research team also provided online tools, facilitating researchers in sequence generation, scoring, and feature exploration. These open resources will promote the development of more innovative research. Overall, Evo2 demonstrates the great potential of large language models in understanding and designing biological sequences. IT can not only recognize coding sequences, but also understand regulatory elements through internal representations, reflecting the multimodal nature of genome language modeling. With the development and application of such models, we are expected to achieve more groundbreaking progress in areas such as genome annotation and functional prediction. That's all for this video. Thank you for watching. If you found it helpful, please like and subscribe, and we'll see you in the next video.